Welcome to our channel. In this video, we are going to see about transcription in prokaryotes. When compared with the eukaryotic transcription, prokaryotic transcription is very simple. Let us see what the process happen in this transcription. Transcription actually is a process through which RNA is synthesized from the DNA template by using the enzyme RNA polymerase, simply called mRNA from the DNA. So that is called transcription. Here the enzyme used here RNA polymerase enzyme. RNA polymerase actually bind to the special region. That region actually called promoter region. Usually promoter is the starting point where the transcription started. And after that RNA polymerase enzyme bind to the particular point in the promoter sequence, it is transcribed and it will reach up to the terminator sequence present in the DNA. So at last the RNA is synthesized from the DNA. Usually these molecular processes are classified as 3 steps like initiation, elongation, termination. During this initiation process, actually the process is started, and during the elongation process it continued, and last process is the termination. For the last process, totally all the processes are stopped. So this is common for transcription, translation, replication. So this is the concept. Here the RNA polymerase involved, the enzyme involved to synthesize the RNA from the DNA. So what is the structure of the RNA polymerase enzyme? In bacteria, it is a very simple system. All 3 classes of RNA like mRNA, rRNA, tRNA. 3 types of RNA actually transcribed by the same RNA polymerase enzyme. Only the single RNA polymerase enzyme. But in eukaryotes, it has some difficult process. RNA polymerase 1, 2, 3 types of RNA polymerase involved in the eukaryotic transcription process. Here, 3 types of RNA synthesized from the single RNA polymerase only. So it's a large and complex enzyme structure. It containing 6 subunits, totally 6 subunits, 5 core subunits, and another one factor is called sigma factor. The sigma factor usually used for the starting of the process, like initiating point. Sigma factor initiating the transcription process. So sigma subunit is weakly bound and can be separated from the other subunits, yielding a core polymerase consisting of 5 subunits, that is 2 alpha, 1 beta, 1 beta dash, and 1 omega. So totally 5 subunits plus sigma factor. So this form of enzyme actually called holoenzyme. Holoenzyme means core enzyme plus sigma factor. Core enzyme means these 5 subunits like 2 alpha, 1 beta, beta dash, and omega. So you just remind that in prokaryotic transcription process, the enzyme is in the form of holoenzyme. It has 6 parts like a core enzyme plus sigma factor. So this is about the RNA polymerase structure. Let us know the reaction step by step. And before going to know about this initiation, elongation, termination process, we should know about what is called promoter. This is a regulatory region of the DNA. It located the upstream region of this gene. It providing a control point for regulated gene transcription. Actually, the promoter has specific DNA sequences that are recognized by the proteins known as transcription factors. Even the polymerase enzyme recognized by this promoter sequence. These factors bind to this promoter sequence, recruiting the RNA polymerase enzyme, and that the enzyme synthesizes the RNA from the coding region of the gene. And another one point, enhancers. Enhancers act to stimulate. Mostly the enhancers are stimulator. It will help to the promoters to initiate the process of this transcription. In common with upstream promoter elements, they can be very active in all tissues or can display tissue specificity. They are modular in structure and can act cooperatively. So it is just helping compounds for the promoters. See, this is the structure of the prokaryotic gene. Promoter sequences are here like a TTG ACA in the region of -35. TAT, Tata box. It is simply called Tata box in the -10 region. Here the transcription starting point is here. So from this side, the left side of the sequence is called -1, -2, -3, -4. That's why -10 region is Tata. -35 is the TTG ACA sequence. Likewise, the right side of the portion is called a downstream. And this left side of the Tata box, TTG ACA box, is called upstream region of the sequence of the gene. So the starting codon actually present in this like light pink color. It indicated in the light pink color. The starting codon is AUG. Usually all the proteins are coded with the starting amino acid of methionine. This methionine amino acid actually coded by the AUG. And center point is the coding region. And the last point is the stop codon like UAA, UGA. So these are the codons actually terminate the process of the transcription. So that is called stop codon. So this is the structure of the prokaryotic gene. The transcription in prokaryotes. So mostly I already said that only it is classified as the three processes like initiation, elongation, termination. In addition to that, one of the extra processes here, template binding. So most notably in the bacterium E. coli, these processes are template binding, RNA chain initiation, chain elongation, chain termination. Let us see one by one. So this is called initiation. In initiation, RNA polymerase, the holoenzyme form of RNA polymerase first bind to the promoter sequence in the region of -35 to -10 region. So the sigma factor, see that the brown color factor is the sigma factor, light yellow color is the RNA polymerase enzyme, the holoenzyme form of the RNA polymerase. So the sigma factor first attached to the promoter sequence, present in the -10 or -35 region. So after the sigma factor bind to the DNA template strand, then only the whole form of the enzyme, RNA polymerase enzyme attached here. So initiating factor is the sigma. So the initiation is continuous. The RNA polymerase binds more tightly to the promoter sequence. Then it opens the bubble. So this is actually called a transcription bubble. Up to certain region, the DNA double stranded are separated as a single strand, because once the RNA polymerase enzyme read the coding sequences present in the opposite strand of the DNA, then only it will copy to the message. Then only the messenger RNA may be formed. So it must be open up to certain period. So this is called transcription bubble. Here the open promoter complex. See that picture. Double helix of the DNA is unwinded here. The RNA polymerase enzyme unwinding this double stranded DNA. So this process actually called initiation. Once the sigma factor attached to the promoter and RNA polymerase enzyme attached to the promoter sequences, this is called initiation process. So this is elongation. Actually, the sense strand of the DNA is 5' to 3' in the region. An opposite strand, that is 3' to 5' region. So that is called antisense strand. So from this antisense strand, it using the messages. Like if suppose the antisense strand have the C sequence in mRNA, the RNA polymerase enzyme add G. Like G present in the DNA, here it add C. So the complementary sequences are added here. It forms a small helical form of the structure. DNA with RNA. You see that pink with green in the bubble, transcription bubble. This green color 5' to 3' representing the newly synthesized form of the messenger RNA. Always the new strand synthesized in the direction of 5' to 3' only. Don't forget that. The elongation of here after the 8 to 9 nucleotides have been polymerized. Maybe the 8 or 9 nucleotides added, attached. Then the sigma factor dissociates from the core enzyme. Because the role of sigma factor is just initiating the transcription process. So it dissociates from the promoter sequence. Then RNA polymerase continues its role. So it adds the opposite sequences present in the antisense strand of the DNA. So just the messages are coding here. The mRNA is synthesized here. So after 9 to 10 nucleotides, it will continue after the decision of the sigma factor. So this process is actually called elongation process. Termination. Termination means almost the process may be stopped here. So two types of termination occurs in prokaryotic cell, like row dependent termination, row independent. Row is just one of the protein. It's a termination factor protein, and this mechanism, transcription, is terminated by the row protein. It is a ring-shaped single strand structure at binding with the ATPase protein. The row protein binds the single stranded RNA as it exits from the polymerase enzyme complex and hydrolyzes the RNA from enzyme complex. The row protein doesn't bind to the RNA whose protein is being translated, because here in the prokaryotic cell, the transcription and translation occur simultaneously. So the row proteins bind the RNA after translation has completed, but transcription is still on. So just the protein is binded to the terminated sequence after the whole process are dissociated here. You just see the picture here. This is row dependent transcription termination. The row protein is attached with this mRNA, and after that it is dissociated all the form of the DNA double helical form and also mRNA. It separated the mRNA from the double helical structure of the DNA. See here, once the row protein bind to this double-stranded helix and the RNA transcript, then it dissociate all the form. The mRNA only separated from the DNA strand, double-stranded DNA strand. After that, the DNA is again, it will form the helical structure. It will rewinding here. See that picture? So once mRNA is synthesized, it is separated from the double helical structure of the DNA. And row independent termination. In this mechanism, transcription is terminated due to specific sequence in the terminated DNA. It may reach the terminated DNA sequences. The terminated DNA contains invert repeat sequences, which cause complementary pairing as transcript RNA from hairpin structure. Because continuously it has some structure like a TTTT sequence, it will attach with each other. Because this is a repeated sequences, it will form the hairpin loop structure. You see that picture here? This is a hairpin loop structure. So once it will form the loop-like structure, the RNA polymerase doesn't recognize the mRNA as well as the DNA, which is not get the sequences from the DNA, as well as it's not bind with the polymerase enzyme. So see that? Exactly. This is row independent transcription termination process. RNA polymerase passes when it reaches a termination site. Then the pass may give the hairpin structure time to fold. See the hairpin structure, the green color hairpin structure. In the second picture, you see that the mRNA itself formed the green color loop, hairpin loop structure. So the fold actually disrupts the important interactions between the RNA polymerase enzyme and also the RNA product. The U-rich region, because the T region present in the DNA means in the RNA the region is U, because it copies the opposite strand only attached here. So the U-rich RNA can dissociate from the template. The complex is now disrupted and elongation is terminated. So this is a row independent transcription termination. Thank you for watching.